This is the Lower 6 Key Assessment 3, which is the core concepts, and this is paper number 1. We've made a help me video, hopefully you've watched the help me video to try and help you do these properly, and this is the model answer. Question 1. So we're given a bit of paragraph there. This is about enzymes, and it's about immobilized enzymes. We've identified the substrate, the enzyme and the product. We do that for any enzyme question really, and then it helps you understand graphs and tables later on. So the substrate is this pectin which glues cells together, which isn't really good for having nice clear juice. The enzyme is pectinase, and then that breaks down the pectinase, and then you get a nice clear juice product. And they are immobilized, they're using immobilized pectinase in the middle. Okay, and if we have a flow rate that is reduced, slower, it says we get increased juice production. So remember that this enzyme relies on a collision with a substrate. And enzymes and substrate, well, not in this case enzymes, but the substrates are diffusing and moving around and colliding with enzymes. And if the flow rate has longer to do that, if the flow rate is lower. So it increases the time where enzymes and substrates can collide. So increases the contact time. Between enzymes and substrates. And in any enzyme question we're probably going to mention, active sites, collisions, enzyme substrate complexes. We want to say those things. So more successful collisions between substrate and active site. Between substrate and active site. Or we could say more enzyme substrate complex is formed. Same thing. So 2 marks there. Then we've got a nice graph. So in the help me video, so I've identified the three enzymes and how they're. So we've got the free enzyme there. We've got the and that kind of representation of that. These are free enzymes that are free to move around. The membrane-bound enzymes, so they're relying on the substrates colliding into them. And we've got the bead immobilized enzymes, where the substrate is going to have to move and diffuse in and collide with an active site. Using the graph and your own knowledge of enzymes, answer the following questions. This is 4 marks. So quite often a 4-mark question, using the graph and your own knowledge might be 2 and 2 marks. Describe and explain. So let's describe the free enzyme, so that's the one that I've done in yellow, at temperatures above 40. So we're going from here all the way down to 70. So what happens, we're going to get 2 marks for this, so what happens to start with? So from 40 to 60 degrees, so from 40 to 60 degrees, this goes down, but what goes down? The volume of juice extracted goes down. So there's a decrease in the volume of juice extracted. So that's our first mark. And then from 60 to 70 there's zero juice. So above 60 degrees, no juice extracted. So here we've done a describing job. And then we need to use our explanation, and it says our knowledge of enzymes. Well, we know that at high temperature enzymes denature. So from 40 to 60 degrees enzymes are denaturing. Or we could say they're denatured above 60. We could say hydrogen bonds are breaking. So that's the tertiary structure, which is losing that kind of 3D shape. So hydrogen's breaking, tertiary structure breaks down. And our problem is, now that our active site shape is changing, it's no longer complementary to the substrate. So the substrate can't fit in the active site. So active site changes shape, substrate no longer fits, or no enzyme substrate complexes can be formed. So this bit is the explain using your knowledge. So that is a typical AO2 type of question, which is using the information and then adding your knowledge to explain what's going on. Over the page. Why do we get a higher yield when using the free enzymes at 20 to 40 than using the immobilized? So that is between 20 and 40. So here. Why is this yellow line, free enzyme, higher than the two other lines? Well, if we look at this diagram here, on these immobilized ones, so these are the immobilized, the enzyme is not free to move. We're relying on the substrate colliding with the enzyme rather than the enzyme moving about as well. And in particular, this bead one, we've got the substrate's got to diffuse in through the gel. Whereas our free enzyme, the enzyme can move and have a larger chance of collision. So that's going to be the basis of our answer. Free enzymes can move. There's more chance of a collision. So remember, this is 2 marks, so we need to say at least 2 separate things. So increased chance of a successful collision, or more enzyme substrate complexes formed. More enzyme substrate complexes formed. Part 3. Explain the difference seen in the results for the enzymes bound with the gel compared with the beads between 20 and 60. So between 20 and 60, so in this area here we're comparing the membrane that are done in red and the beads done in blue. So what we notice is the red is always above the blue, so we're getting more volume of juice extracted using the membrane compared to the beads. And this is not too surprising, as we can see here the substrates, they just need to collide with this membrane. So they're surrounding this membrane, just need to collide, whereas with the beads they need to diffuse through this gel to get at the enzymes within. So of course it's going to be slower. So the membrane bound enzymes are directly exposed to the substrate. Remember 2 marks, so we need to say 2 things. We said 2 things there. And again we're going to say 2 things. We're going to say with the problem with the beads. So with the beads, the substrate has to pass into the bead or diffuse into the bead. Diffuse into the bead first. So of course it's going to take longer. And the last part, which is 4 marks, we have to say 4 things here. So this apple juice producer wants to increase their yield. Use the information. So use all the information to outline, so we don't need to give a lot of detail, outline how they could adapt the basic method shown, which is that, to extract a higher volume of apple juice. Explain the recommended modifications. Right, we want a high yield of juice. High yield. Where's the best yield? So the best yield here is this. That's our best yield. So whatever we're doing there, I'm going to suggest that. I'm going to suggest that we do it using whatever that red line is on there.
Yeah, I thought you were going to teach.
So you're going to use our red line on here, and we're going to use a membrane, and whatever the temperature is there, we're going to use that. So membrane and 60. And what else are we going to do? We need another two suggestions. We need how could we increase this membrane? Well, you know about exchange and things like that. Well, let's just have more membrane and more surface area. And we need a fourth thing. Well, so we talked about here increased juice. What was going on there? Reducing the flow rate. So that's my 4th thing I'm gonna write about. So first thing, use the membrane bound enzyme. Second suggestion, use 60 degrees C. Yeah, and these two had the highest yield. So membrane bound, that was the highest. yield 60 degrees. That was the highest yield. And so for this membrane now, what could I do? Use many membranes to give a large surface area. Really difficult thing to say, that. And it sounds easy now with when you're reading the mark scheme, but difficult to think of in the exam. And fourth, when you're stuck for ideas, always read the question again. Use a slow flow rate for increased contact time. So use a membrane, use 60. We could give a little bit of explanation there. We've got another line, so we could have said, higher kinetic energy, highest yield, more kinetic energy, possibly said that. We've got more surface area. Use a membrane because it's not denaturing. So there's our kind of 4 suggestions to get 14 marks. Quite a difficult question, that.